So, hello everyone, and welcome to today's open day chat about the MSc program, which is Nanotechnology and Regenerative Medicine. My name is Connor Proctor. I'm on this course currently, so I'm halfway through, and we will be joined today by Gavin Jell, Professor Gavin Jell. So he will go through a presentation with you guys, give you a little bit of insight about the program, what it offers, what we'll study, and things like that. So we hope this session will address any of your questions that you might have. So I would imagine you probably had a look at the program and you may be interested in it. So you'll be able to ask myself questions, you'll be able to ask Gavin questions as well at the end. So hopefully we'll get some insight into hopefully one day you studying your master's program with us. So I'd like to pass it over to Gavin now, and we can go through the presentation.
For a lovely and professional introduction. You can definitely do this again. I thought maybe we'd start off by asking some questions about you. You're on the course. I think it's interesting for the students to know a little bit about why you're doing this course. So I'll start off with some questions for you, if you're happy.
Of course.
So why did you choose this MSc program?
So I chose this MSc program. So I have lots of friends actually within the tech industry, and I have a background in the medical industry. So as a biomedical scientist, I worked in PCR for a few years. So I was, especially during COVID, I was very busy. And I wanted something which I was going to challenge myself and also something which was kind of new. And I believe that nanotechnology and regenerative medicine seemed like kind of like the cutting edge of the science in terms of medicine. So really, it was to enhance my knowledge about medical applications and sciences, really. And that was a big. I think it was more like the. Okay, I have another personal thing that happened to me. I broke my leg very recently, and they put a metal rod in my leg. And what I found out was actually through what we will study, it's to try and create a treatment or through a scaffold to try and naturally help that regrowth process. So through regenerative medicine. So there's lots of different probably inspirations that got me to where I am. But yeah, that's a few of them maybe. So
We'll talk a little bit more about those technologies in the session.
yeah.
But I wondered, like, it'd be interesting for me to know, um, do you think your perceptions of like nanomedicine or regenerative medicine have changed from before you start the course and now that you're in the course?
1,000,000%. I think there is always that futuristic aspect where you think we're going to shrink down little people and that they're going to start coming into your body and maybe repairing something. It's not necessarily true. And I think the biggest kind of thing, which was I didn't realize, was how much interdisciplinary goes into it. So how much physics we might have to look at, or how much we actually are paired with clinical um, so with doctors, so the clinical side to it. So I think there are the misconceptions of it that we probably see in media or in books, but it doesn't mean it's less glamorous. It just means that we will have to work a bit harder to get to that point.
Thank you. I know that I talk a lot about like the hype in the regenerative medicine, stem cell therapies. There's lots of it in the media. Printing 3D organs is,
Yeah,
you see everywhere, and it's always in the next 5 years. And we talk about that in the course. And I think it's, what we try and do in the course a little bit is to narrow down the challenges that are there, so that we have, realistic expectations of what we can do and to move the field along.
yeah.
No matter where you go, it's in that development, that criticality and understanding of the field as a whole is important. Uh, what's your favorite module, Connor?
So my favorite module, and I promise he didn't tell me to say this, but is tissue engineering. Oh, hi, Raza. Welcome to the open day. But it is tissue engineering. I think this is probably one of my first real insights into the different ways we do tissue engineering, the many definitions that actually are out there for tissue engineering. And if you do eventually come onto this course, and you do get taught by Gavin, a big tip is to research the definition of tissue engineering. Um, but yeah, it's tissue engineering in general. Yeah, so the different methods that we can do it, what goes into it. Um, I think recently I've been also been having lectures on biomaterials, and this is something again I've have no real previous experience with. Um, but it's so interesting to understand how these two materials can either be incorporated with each other, or how we can use them separately to then lead on to something else. Yeah, it's amazing, and the different methods we can then do it either by through different gels or whether it be 3D printing. Yeah, it's amazing. So
Brilliant. I'm pleased you said my module, obviously. And what do you think the biggest difference is between your undergraduate study and your postgraduate experience in the masters? Can you name some differences that you
yeah.
found or experienced?
Yeah, interaction with my supervisors, or with PhD students. I think everybody is always very willing to help, and everyone's very knowledgeable. It is a bit of a jump up, of course. You know, you're doing your masters, but the research you feel like, for me anyway, I feel like I'm doing real research. Whereas in my undergrad, I kind of felt like it was a very I don't know. Maybe I shouldn't say that, but it felt like it was more. I was writing a paper, and that was that.
Abstract, maybe more
Yeah,
abstract. Not, I think, one of the things we try and
yeah,
do, not just in our course, but across like the Faculty of Medical Sciences, is that, we have, very good researchers, world-leading researchers, but also everything is really kind of directed in terms of patient need. So you can see, even if it doesn't seem immediately relevant, you can see the pathway of how that would help
exactly. Yeah,
patients or medical benefits.
yeah. Translational. I think that's very important. And I think from my perspective, from what I want out of this is that my work can potentially be translational, and I can potentially help others in the future. So yeah.
So, following on that question, what are your
Mhm.
plans for the future? Do you have firm plans in terms of what you're going to do next in
I
your?
do. I do. I would like to do my PhD. Unfortunately, time has passed at the moment for me to apply for my PhD, but I don't necessarily think it's a bad thing. I would like to have a year in industry, pick up some more skills through industry, and then look at applying for my PhD. I can still do that now, and I'll probably look to do that in the future. I'd like to keep within either biomaterials and regenerative medicine. But that's really my end goal. And I would like to teach. I already teach in my spare time, and I would like to teach academically at universities. So yeah,
So you want to be an academic?
yes, I
Excellent.
would like to be
That's
academic.
good.
Yeah,
I think the most common like career uh, kind of route for our MSc students is doing a PhD. So a lot of people do the course so that they can build their CV to make them kind of be able to get the PhD in funded positions, and lots of our students do get those funded positions. Um, it's obviously much easier for funding if you're a UK student than it is overseas. There's not impossible, but there's different challenges in terms of getting that PhD funded. Good. Well, in that case, I will crack on. I've got some slides that are about the course. Um, but as Connor said, if you have any questions at all, for me or for Connor, please put them in the chat and we'll address them when we can. I'll keep asking you questions, Connor, throughout, if you don't mind,
sure,
just
no
because.
worries. I can.
Well, okay. So I'm going to share my slides. Um, oh, I don't want to be there. Excuse me. Wrong screen. That is an interesting screen, but the wrong one. Okay, sorry, bear with me.
So again, if you do have any questions, just don't be afraid to put them in the Q&A section at the bottom, and then we can kind of go through them and answer them at the end as well.
Sorry, a little technical issue. I don't know why it's stuck on that slide. Let me go back. Might just save that. Yeah. Should work now. Okay, we are ready to go. Okay, so I'm going to give you a little bit of an overview about the course. As Connor beautifully introduced me, I am the director of the program. My research is directly in the area of the program. The program's been around for, I think this is 13th year. And kind of overall, we're looking at tissue engineering, nanomedicine, and biomaterials. And a common theme is understanding what happens when we put materials in the body, and can we have improved materials interactions? And that could be materials which can treat cancer, so nanomedicines, or it could be uh, improved orthopedic implants or improved breast implants, or it could be materials which do more in terms of promoting regenerative processes in the body. And we talk about all those different areas, and this is my research area as well. Um, So there's lots of new medical innovations that have existed. There's lots of hype about these innovations, and possibly you could argue that some of the translation in some of these areas hasn't been as great as anticipated originally. But we talk about tissue engineering, stem cell therapies, nanomedicines, and biomaterials. We have a team of really excellent teachers, educators, and researchers, and they are experts in these different areas that we discuss, that I've just discussed. There are 5 taught core modules: nanomedicine, applied tissue engineering, biomaterials, research methodologies, and then there's a practical module where you're in the lab to learn some basic techniques in terms of cell culture and characterization of cells, which will help you with your project as well. And then there's three optional modules, which you can choose one of. There's a translation module, which discusses about how you can translate these medical innovations into commercial products that can benefit patients, stem cells in regenerative medicine, and advanced tissue engineering. We have a research project, which slightly different to lots of MSCs. It's 50% of the course. So we really want research to be like the focus of this MSc. There's lots of reasons why we have 50% research project, which you can ask me out later. But the idea is that we can engage you in the research in term 1, and you keep that kind of research project as an overarching goal, and the taught material helps you with that research project. So if I give you like a brief overview of the year, term 1 is most of the core modules, heavy teaching. Term 2, optional modules plus 1 core module, and you should start engaging with your research project now. Connor is in my research group. He's engaging with his research project, so he's you've had some lab time, but also in terms of the background research as well. We try and allocate the projects in term 1, so we'll give you a research booklet, and then it means that you can do some background reading and be engaged with the project in term, as soon as you come back in term 2. And then the third term is all research project. Just to briefly go over some of the modules. So we have nanotechnology and medicine. This module discusses how nanomedicines can benefit the diagnosis of disease, have more sensitive disease, better imaging of disease. It talks about targeted drug delivery. Can we create nanoparticles that are labeled so that they can go to particular disease sites and release the drug there. So we have a higher concentration of the drug where it's needed and reducing systemic effects. And then there's also a bit about tissue engineering. So how can we use these nanotechnologies, nanoscale biomaterials to help direct cell behavior for more regenerative purposes? Um, like all the modules, I think we all start with discussion on what actually the topic is, because I feel this is important, the defining what we are, because in the public domain, there's lots of misinformation about what nanomedicine is, for example. It's not creating tiny robots. We can't create tiny robots. It's not uh, it's not possible in terms of the dimension of atoms and how big they are. Um, but what we can do is to create nanostructures to direct cell behavior or have targeted delivery, keep drugs or vaccines alive for longer inside the body, and use this technology in ways which we haven't done before to improve the diagnosis or treatment of disease. We talk about, so this module is led by Kate Ricketts. She is a physicist. So she talks about the physical reasons of why size matters, the physical properties. Um, and also we talk about applications. Lots of research in nanomedicine is focused on cancer treatment. So trying to get your medicines to difficult to treat cancers. But also, one of the biggest successes in nanomedicine is in delivery of RNA. For example, for the COVID virus, where you have your mRNA, which is um, encapsulated in a nanoparticle, in this case, a lipid nanoparticle. If you have a Pfizer vaccine, for example, and that means that the mRNA doesn't get destroyed as soon as it's put in the body and keeps the mRNA safe until it can reach the cells. As I mentioned, targeted drug delivery is a really interesting area of research. Currently, there are no targeted drugged nanoparticles. So there's no nanoparticles which have a particular label on it, which can be directed to a particular tissue. Uh, and that's despite 25 years of research in this area. We do have nanomedicines, but we don't have targeted nanomedicines. And what I talk about in this course is the challenges of why that is. I have some research looking at how nanoparticles interact with the protein corona, so non-specific proteins. And I think that's one of the main reasons why we don't get the nanoparticles to where they're needed. And this kind of like concept of like, targeted nanomedicine, we discuss about all the different barriers that are present in the body to stop our nanoparticles from reaching the tumor site. We also talk about other applications for nanomedicine, such as diagnosis. I won't go into this too much, but one of a property of gold, for example, is that if it's a larger particle, it can be blue. If it's a smaller particle, there's the color change in red. And we can utilize those properties for diagnosis purposes. And this is just a list of different lectures which are in this module. And the assessment, they have a poster assessment about research and also an examination, which I'm sure is fun, Connor. My module, which is Connor's favorite module, is tissue engineering. This is good. This is really a talk about what happens when we put materials in the body and how can we change a material's properties to direct tissue regeneration? How do we make new tissues? We talk about different approaches in tissue engineering. So trying to make tissues, new tissues outside the body, which I think what most people are more familiar with. So growing bone, which is my area of research, or other tissues outside the body and then implanting it. But also in situ tissue engineering is where we have a material, a tissue scaffold, and we put that directly into the body and the properties of the scaffold will induce new tissue formation. We talk about decellularized scaffolds, which is a hot topic at the moment. Decellularized scaffolds are organs, for example, which here we've got some livers where the cells are being removed and the patient's own cells will be added so that we don't have an immune response. This is another strategy in tissue engineering. We talk a little bit about xenotransplantations as well, and why it's not feasible, despite earlier, I think it was last year, there was a heart transplant where they used genetically modified pig heart and planted it into a human, not very successfully. So meat is another area. So meat that's been generated from muscle stem cells, where you don't have to kill the animal, but it can generate meat. And we have a few researchers who are interested in this area of research, are developing ways to scale these meat production up so that it's more commercially viable. You can already buy chicken nuggets, which are grown from stem cells, but they're super expensive. Another area of tissue engineering which we're interested in developing is trying to just create better models of disease in vitro. And to do that, we need to have tissues that more accurately mimic the native environment. So this is Denise's research, who was a postdoc in my group, and she was able to create bone in 3D hydrogels, not just any bone, but cancerous bone. And this was really interesting because we wanted to study particular bone cancers. So the assessment for this module is slightly different. We're trying to really focus on critical analysis and having a deep dive into uh, uh, what's good research and what's bad research. And from learning what's good and what's bad research, it means that that can inform your own research practices for your project. As Connor mentioned, the biomaterials is another module where it's talking about the different types of materials that we use in medicine, with the aim of understanding how material properties influence the outcomes of those implants. I think, one of the big problems is about implant failure. I take the view that all implants fail, and soft tissue implants will have much quicker failure than hard tissue or bone implants. But breast implants will have to be removed after 10 years. The failure rates of other soft tissues is much higher. So this module talks about different types of materials, how their properties, physicochemical properties, relate to their function, and developing new materials. So as Connor mentioned at the start, we can have composites, nanocomposites, which can marry different material properties together to have a better material that hopefully has lower failure rates. As part of this module as well, we talk about characterization of materials. So if you're making nanoparticles, for example, or you're making a new material, what's the best way to understand the mechanical, surface chemistry, etc., which I think is really important for both the research projects, but understanding the literature in this field. All our students do a research methodologies module. This is joined by lots of other students on different courses, and the idea is it gives you the philosophy behind developing testable hypotheses, but also making sure that you're aware of the best ways to do statistical analysis. That's not very useful for you. There's a practical module. The practical module is in term 1. It's a nice module. I think everyone enjoys it. Connor, do you like this module?
Yeah, it was useful for
What did
sure.
you get out of the module?
Actually cell culturing. So I'd never cell cultured before, which was really handy. Um, and it's something that you're most likely going to do in your research project. I mean, again, depending on where you go, but at some point I'll have to be doing some cell culturing. So if you haven't had that experience before, it's really great to have. I had experience in using PCR previously, but it's not the exact same way. We may use all sorts of different types of instruments again that I never had a chance to use even when I worked in industry previous. So I think it's really useful.
Yeah, just like Connor said, I think the course is fairly unique in that we want our students to be able to do the cell biology, the basic cell biology, cell culture, the cell characterization in terms of gene expression or protein expression, but also material science and physics. So it is a properly interdisciplinary course. Most of our students are from a biomedical background, but we also have medics, surgeons, and physical scientists that join us as well. And we're not specific. We don't say we have to have a biomedical background to join the course.
Like for example, within my research at the moment, I'm working alongside two medics, so both of them being plastics and burns surgeons. So that's
Thank you. Some of the optional modules. So one of the optional modules is translational research, where uh, it's like a dragon dense pitch where the module lead, Dr. Lee, tries to get you to think of ideas and then assess these ideas, and then also start thinking about how you translate these ideas. So it's development of a business plan. It's quite a fun module, very hands-on. The research projects I wanted to mention. So the research projects, as I said before, are 50% of the entire MSc. The allocation process is that in the first term, you'll get a research booklet of different research projects. You will look at that. You'll talk to the project leads about their projects. You'll pick your favorite three, I think, and then they'll be allocated based on the project supervisor's choice if there's a clash. I think I can't remember this year, but it's rare that there is kind of too much overlap in terms of students wanting to do the same project. Connor, do you want to add anything to this?
um yeah, I mean there's so many different options that you can go or that you can look at in terms of your research, whether you're looking at going along with Gavin's research and looking at bone regeneration, or maybe you might look at creating a hydrogel to mimic certain disease models, or use it as, to then create certain disease models. There's so much that you can do. You can look at different systematic reviews. So there's lots of different types of research, and yeah, I don't think there was too much overlap, like you say, from when people were applying for
What
what they wanted.
I guess what we're saying is that most students get the projects they want to do,
Yeah,
and quite often you are because you are our students. You're quite desirable because you've been taught well, but also because your projects are longer than a lot of the other projects in the UCL. It means that the PIs, the group leads, can get more out of you. They have time to train you and get results from you, and that's what we want because this is real research. And the reason the group leads are offering these research projects is they want real data for publications, etc. So this is a rough overview of the kind of term one. The practical module, you have lectures and workshops, and that kind of runs, it's like a fat, short module at the start of the term. And then you have biomaterials, tissue engineering, and research methodologies, which are core modules. There are some co-curricular activities. We are aware that we have kind of a mixed cohort of students. So we have these basic biology workshops to especially initially they were designed for medics in particular because the medics don't know much about cell biology, which we think is quite important. Most of the material science and physics knowledge is taken from a point of view of that we have to teach you everything. So it's quite a lower level of entry. Um, what else was I going to say about this? Uh, so we have these basic biology workshops, and then term two, we also have journal clubs, which I'll talk to you about in a minute. And then term two, it's more research project, optional modules, and one core module. As I said, these workshops, they're really led by our PhD students, we call them postgraduate teaching assistants, and they're kind of led by what you want to learn about. But we think some of these like basic cell biology topics are useful. It's also quite good for students because it's very interactive in terms of developing professionalism, engaging with the PhD students as well. There's also a basic chemistry module as well, which we have online. And we also have a journal club. So this journal club is where students will present a paper, um, a research paper to their peers. And this is aligned with some of the assessments. Connor, do you want to say anything about the journal club, how you found it, whilst I plug in my computer?
Yeah, sure. So the journal club is really, really useful to really critically analyze papers. It's a skill which you have to work on, and it's not something that that came easy to me or even to others, and it's something that you do have to continuously do. And I think being able to have a look at a paper, um, clearly see what their hypothesis is, clearly see what their results are, what they're actually trying to present is so important. And journal club, or having a journal club amongst your peers who have different ideas on maybe or criticism, should I say, about a paper is so vital. And when it comes to real research, it's stuff that you're going to be doing potentially even in your research projects and your later career. So it's really useful.
I think it's quite an eye-opener to students how poor a lot of publications are.
Okay.
And it's something you want to be, like we try and get you to be really critical, but it's only by knowing what poor standards there are in some publications already that you can actually do good science. And that's the aim. Recognizing what poor science is so you can do good science. Um, Good. As we mentioned earlier, it's a very international cohort of students from different backgrounds. As I said, most students are kind of from a biomedical background, but we have medics and physicists, material scientists, etc. on it as well. Some chemists now and then. And a mixture of home students and overseas. I think we have more overseas students this year than home students, Connor. I don't know
Yeah.
how
I
many.
I f- I,
You're
f-
a home student.
I think I
This year.
think there's maybe two or three
Two home students,
potentially.
three.
Yeah.
Okay.
So yeah, there's
It
not
does vary a bit between year to year.
yeah. But it's great because I mean you again you just broaden your horizons and who you talk to and your connections and networking, and yeah, it's great.
So in terms of delivery, we have lectures. We also have a lot of online learning. We use a particular portal called Moodle. There's some like quizzes and things to do on there as well. So I should mention that we are based at the Royal Free Hospital, which is in Belsize Park. It's about, well, if you want to walk, it's, I don't know, a mile? I don't know how far it is from UCL, Bloomsbury.
Uh, what, the main campus or
So,
of
so
Royal
I,
Free?
I I cycle everywhere now on these on the
Yeah.
bikes. It takes me, it's about 15 minutes on the bike, but it's
Yeah.
obviously longer to walk. If you go on public transport, it's about half an hour, isn't it, between the
Yes,
campuses?
it's about half an hour, maybe 45 on the bus. So if you take the bus around 45, half an hour on the Northern Line. So yeah.
But our kind of department, our where we do our research, most of the research projects are based on the Royal Free. I think all the lectures, apart from the practical module and the research methodologies, are based on the Royal Free. The other thing I think we're trying to do in your year here is to build your network. So we want to encourage you to engage with the wider network of researchers in this field. So I'm a trustee, for example, in the British Society of Nanomedicine. I'm really keen on students to join that. It's free for them, but it connects them with supervisors, the experts in the UK on nanomedicine. And if you want a PhD, it's likely that one of them The people that are members of nanomedicine are the people that you're going to work with. Careers. So what do our students do afterwards? As I said before, the majority of our students end up doing PhDs, but not all the students do that. Some go into the more kind of medical innovation and translation uh, area. So that could be in terms of startups, biopharmas, or there could be more research environments. That could be either for small or medium-sized enterprises, but also larger, uh, big pharma like Glaxo Wellcome. So there's a real mixture. The medics obviously continue their medical career. Um, so there isn't a particular area that our students do, and it really depends on what they are interested in. Yeah, I'm happy to answer questions about that afterwards. But that's it. That's the talk. Uh, we're going to be discussing your questions now. So if you have any questions, put them in the chat. Or, I'm not sure if it's possible, you can ask us by putting your hand up. There is some questions in the chat, is there? Well,
There
one
is a
few
question.
questions.
Yeah. Yeah. So I think there's one more. So the question we have in the chat is, I was wondering why the course Nanotechnology and Regenerative Medicine lasts 1 year, whereas other STEM postgrad studies last for 2, especially when it comes to medical sciences. Isn't there more content that needs to be learned from both a medical and technological standpoint, and how the two interconnect? Okay, so did you want me to answer it first and then maybe you, Gavin, or it might be more of a better for you? But
I'll answer the question. I'll answer the
whichever.
question, shall I? And then you can tell me
Sure,
if I'm right. So in the UK, most masters
sure.
are 1 year, not 2 years. That's full time. You can do the course part time, which will be over 2 years. Um, uh, or you could do it flexibly, which is over 4 years if you wanted to. But the full time course is 1 year. In Europe, in some universities in, I know for Japan, because I'm a professor in Nagoya Institute of Technology there. Their masters are 2 years. So it does depend on the system you're in, or the education system, but it is the norm in the UK to have a 1 year master's. If you're doing it part-time, if you're working, then you do it part-time and it takes longer. Connor.
Yeah. Yeah. I mean, that's basically what I was going to say. I mean, I think
Hmm.
also they are more intense generally. Speaking to people who I know who've done their masters, and I do believe in the UK that they seem to be slightly more intense than maybe doing it in the States and elsewhere. But again, that's just based on people I've spoken to who've done either their degrees abroad or in the UK. So yeah.
It's 180 credits, which means it's supposed to be the 12 months. So if you register when do you register? Kind of end of August, start of October, and then it ends the end of July, and then there's a month for like marking your thesis and vivas, and then you're done.
Is there any other questions? Yeah. Cool. So Daniel, so I have a question for me. Okay. I wouldn't want to ask about a private topic, but in what areas are you thinking about going into in industry this next year? So really, this can open you up into lots of different areas. Um, I would ideally like to stay within either biomaterials. Um, Oh, am I still there? Yeah. So I ideally like to stay within biomaterials, um, or look at a lab tech position, because I do have experience using, or I will have experience using 3D printing or bio 3D, well, yeah, 3D printing, and making these different things. I'm ideally that's what I'm looking to go into. Um, but I'm also looking at biomedical science roles, which I've done previous. I also do think it's very useful to network, as we mentioned previous. I lived in Korea for a few years, and I'd network with a good few people over there, and so there's always potential positions within these biomaterial companies or things like that. But it's it's a very good idea to network, and especially if you are in London on this course. There's so many opportunities to network with other people. So I think it would be mostly going into chemical labs or looking to go into something to do with stem cells. I think personally for me, I would want to go into materials, biomaterials, and then looking for my PhD again. Would like to go back into regenerative medicine and using materials again.
I mean, I would advise Connor
Hopefully that answered
here,
the question.
yeah. Um, I think I would advise Connor that, obviously, there is a challenge in terms of getting a funded PhD, and there is kind of a bit of a yearly cycle for the doctoral training programs, which is why, a lot of the deadlines have passed for this year for September entry. But there are other charities which are open all year round, which you can look at as well. Of course, like you want to make sure, I always advise the students, this is a year to try and build your CV to make it as good as it can be. Don't make your network, try to get presentations. I'm sending Connor to a conference, the UK Society of Biomaterials, um, uh, where he'll present his work, which would be good for his CV. So it's those kind of career building activities which are important, networks you build, activities, evidence of
Yes.
engagement and success in your field will help. Um, I think if you like research, you like research, and I think like there's always a difficult conversation about whether I should do a PhD or not. I think if you're interested in academia like Connor, then there's no question you have to do a PhD to become an academic.
Yes. I think from my own perspective as well, I love learning. And it sounds cheesy to say this. I'm an older student. I'm actually gone into I'm now 30. I'm now in my 30s. And I'm now doing my master's. So I've had life experience of working different positions, different jobs, and I've always come back to more of the medical science sort of fields. And this is kind of what I want to do. Um, so yeah, that's my justification. So we have another
So,
question. So is
yeah,
there a chance to do? Oh, yeah, sorry. Yeah.
yeah, there's a bit of a lag, Connor, on your feed. I don't
Oh,
know if it's um.
oh, is there?
So yes, in terms of yeah, so there is certainly the opportunity to do projects which are in terms of synthesis of materials. Lots of our um PIs, group leads are researching this, and certainly hydrogels, polymers, nanocomposites, and on different scales. We have a nice new bit of kit in nanoparticle synthesis machine, which is uh, uh, one of our PI's new toy, which is fantastic. So there is lots of opportunity to synthesize new materials and characterize them,
Yeah.
because
So
that's obviously the,
y-
the important point.
yeah, I mean, currently I'm working with polymers and bioactive glass. So I will actively synthesize different, a particular polymer, um, do biochemical characterizations, mechanical characterizations. We even use the SEM. And me again being a bit nerdy, I really really enjoyed it. Um, it again, yeah, it's I guess it's one of those sort of things that you always see in pictures or you see online, but actually being able to see what you've created, so the synthesis of a your polymer scaffold, for example, and then look at it through the SEM, and you can see how the minor differences within the surface, morphology, right word, yeah, yeah, the surface morphology is yeah, it's just incredible in my opinion because it's so small, so small.
We do have a quite a nice imaging suite, which is relatively new, but it has some electron microscopies, so transmission electron microscopy, and also scanning electron microscopes.
Yeah.
Um, but yeah, so it's quite, we have some nice imaging techniques available to us, which is good. Some good characterization techniques. It's actually fairly rare where you have kind of the polymer synthesis. So we make the materials, we characterize the materials, we put cells on the materials, all in the same kind of space in the same building, which is in terms of, unusual, I think, rather than the material science building, making the material, and then sending it to the life science department to do the tests on those, and then imaging them in another department.
And then with Gavin being my supervisor, we then come and say, okay, this is what we've done. And then as Gavin's job is, did you test for this? Did you test for this? Did you test for this? And you're like, oh no, we haven't done that yet. Maybe we should go back and then test for these other things. But that's
It,
the
it
process, right?
yeah, it's a good point in terms of the group like supervision. There is differences. I'm kind of I have PhD students who go in the lab. I rarely go into the lab, so the PhD students are the one that are really supervising Connor, and then I'm the overarching kind of one my lead. So it would be like, I'll be the one that will be helping directing the research and then helping them in terms of the thesis writing and the area for PhDs as well as the MSc students.
Yeah,
But the day-to-day supervision is largely led by either PhD students or postdoctoral researchers who are in the lab all the time.
yeah, you're
Um,
the maestro.
yeah.
But yeah,
There's another question.
is there any other questions?
Oh,
Oh, I can't see on my side. Oh,
how
I
are we doing
can't
for
see the
time?
question.
Well, I think we've got another. Is it supposed to be an hour
Yeah, we've
or?
got another 10 more minutes. So we've got another 10 more
Um,
minutes. So yeah, if you have any questions, it can be about anything. So it can be about the course, it can be about my own experience, it can be about if you have any fears with anything. Even if it comes to if you're an international student, um, is it accommodation or funding or anything like that? So next one, do you also focus on advanced nanoscience as well, such as the development of spintronic devices and their possible implementations for medicine? Spintronic.
So we have a good cluster of nanomedicine researchers in our department. So some of them, like Kate Ricketts, who's the module lead, is interested in nanomedicine, nanoparticles for enhanced radiotherapy because she's a radiotherapist. So to whilst other ones, Hirak, I keep calling people their first name. I should call them professors. So Hirak is interested in nanoparticles, but also in terms of drug delivery, but also in terms of regenerative medicine. So releasing growth factors for regenerative medicine. I'm interested in understanding how nanoparticles interact in biological environments and can we change that? So kind of more of a kind of modeling approach and having better in vitro models of nanomedicine to hopefully improve the translation of those nanomedicines. Kind of think of other examples. Oh, there's Nicoletta, Professor Nomaku, I should say, who is very involved in cancer nanomedicine, and she's developing responsive nanoparticles that when they're in the tumor microenvironment, they'll change their properties, so they'll release a drug, etc.
Is that Professor Boyd is doing a disease model?
Oh yes, that's right. So also that there's also uh, John Council, everyone's who's looking at using nanomedicines to deliver genes in cells in different ways. So that's also interesting.
Yeah, cool. So next one, so from Daniel, is there any type of assistance with accommodation in the harsh London market? So, from my standpoint, London can be very harsh, but UCL does offer some good accommodation options. You can also, if you are coming from abroad, there are scholarship options. So if you, I'm not sure, I think this June, a lot of the scholarships where they end. So I would really recommend is applying for some of these scholarships if you're coming from abroad for accommodation. Um, in terms of Gavin might know a little bit more in terms of assistance. As a home student, I have the benefit of staying at home. So I live in Norwich and I commute in, but that's because I can live with my parents. So that's kind of lucky for me. But yeah.
Yeah, so I think there are that I'm aware there are forums available where you can look for flat shares, etc. I think there are halls. I know UCL is building more halls to accommodate students, but they do offer assistance in terms of finding accommodation. I think most of our MSc students are not too far away. The ones that have got halls in Camden or Bloomsbury. Um,
Yeah. Islington. So, or I say Islington, Highbury and Islington, Camden. Someone as far as Richmond, which is not, it's not super far, especially if you're going to be based at the Royal Free. So you do have scope to live wherever. So I think everything's pretty close.
Miliana's just put some links in, which is very useful. Thank you, Miliana.
Yeah.
I
Any
think,
other
like,
questions? Yeah.
yeah,
Oh,
so I'll just
go on.
say, like, obviously it is worth considering. It is expensive. London accommodation, London is more expensive, and it is worth looking at the total price cost uh, whilst you're here. There are some scholarships as well. So if you wanted to look for funding awards, there's Miliana's put in a link there as well. That are different funding schemes that are available for students.
Yeah. So was there any other questions? Oh, got one coming. So are typically all research projects done in research groups from UCL, or is it possible to do them outside?
Um, they're all done in UCL. I'm saying this, there have been exceptions where students have done in hospitals, so they've been led by, but it's always so they've been no, there's been surgical projects which you've had, or more surgical leaning, which have been led by a surgeon in a different hospital that's not associated with UCL. But in terms of our standards and making sure that our supervisors are aware of the marking criteria that we're using, what the thesis standards we expect, everyone has to have a UCL supervisor as well. So there has to be at least one UCL supervisor for your projects.
And I know, for example, for my research, we are collaborating with another university, but we don't necessarily, they're not doing our research or anything. We just collaborate with them to use maybe some facilities or something like that. So we don't do the research with them, but we may collaborate in terms of using equipment or something like that.
So particularly for Connor's project, I think we're getting our glass particles are being manufactured by a group in Germany, sending them to us, and then for ICP, one particular characterization technique, we go to Imperial College to do the analysis with another collaborator. So, you do get to see there is some collaboration, but they are largely based at UCL.
Yeah.
Well, good.
Okay. So if there's no more questions and everyone's happy, are you happy, Gavin?
I'm super happy.
You're super happy? I hope everyone else is happy. And with what we discussed today, this meeting will be recorded and it will be placed on the UCL website as well. So if there is anything that you maybe missed or you came a little bit later, you can always catch up on there as well. And yeah, if there's nothing else, we can then leave it
Uh,
there.
Good. Well, thank you so much, Connor, and thank
No
you all for
worries.
attending. And please let us know, if you're in London and you want to come and visit our labs, you're very welcome to. You'll see us in person. I know it's a big decision. Or if there's any other questions, please email me, and I'm happy to respond.
Cool. Okay. Thank you.
Bye now.
Bye.